All right, it's time to start. 2:30 sharp, and I can see that we have people joining. So let me introduce for today Axela webinar Sebastian Clerc. Today he will explain us a bit of Cellenion solution for sorting of cells and spheroids using, in fact, the isolation method developed by Cellenion. So, Sebastian, I give you the word, and for all the participants, enjoy the webinar.
Thank you very much for this kind introduction. Good afternoon, everyone. My name is Sebastian Clerc. I'm working for Cellenion, and yeah, let's go ahead. I will present you the solutions we have for single cell spheroids and organoids. Just a few words about who we are. So Cellenion is a subsidiary of Cellenion, and we are together part of the BICO group since 2016, whose mission is to combine biology and technology to develop groundbreaking technologies for applications in biology and medicine. We, Cellenion, were founded in 2016 in Lyon, in France, and we are a subsidiary of Cellenion, which is based in Germany and which is a leader in precision low-volume dispensing technologies. And what we do at Cellenion is that we leverage those precision dispensing technologies in order to handle biological objects. And so we are experts in single-cell solutions in various fields with applications in cell biology, molecular biology, microbiology, sequencing, proteomics, microscopy, etc. Our mission is to provide technologies to research institutes and companies for them to better understand and master cell biology. What we do is that we provide instruments, first of all, instruments that will be able to sort cells or cell aggregates and dispense them one by one with high accuracy. And those instruments are also able to dispense picoliters and nanoliters of liquids. In addition to that, we have some consumables so that we can provide turnkey solutions for researchers so that they can run complete workflows on our instruments. And we also propose some services from experimental design of the experiment to the analysis of the data. I will tell you more about that. So just as an introduction, but probably you're already well aware of that. Why do we want to work on single cells? Well, first is to understand the cell heterogeneity within a tissue or a tumor, for example, in order to understand which cells may have different properties and characteristics from the others. And this is something that we cannot identify when working in bulk. I do an analogy. If you have a fruit salad, you are able to pick every single fruit and tell this is a strawberry, this is an apple, etc. But if you mix and do a smoothie with those cells and want to identify in bulk, you may not identify all the fruits that were in your starting fruit salad. And so overall, this enables researchers to better understand cell biology and diseases, and in the end paves the road towards personalized medicine, choosing the right treatment for the right patient for the disease one patient has. The second main application is the production of drugs and testing those drugs for toxicity and efficacy. A few examples are the production of monoclonal antibodies and immunotherapies, for example, to fight cancer, gene editing as well, like CRISPR-Cas9 that is more and more used for cell and gene therapies, and more generally working on stem cells in order to develop better preclinical models on which we can test drugs and ultimately complement and replace animal models. And so what we have at Cellenion is that we have developed a range of instruments in order to provide solutions to researchers in that area. We have two instruments. First on the left, the CellenONE, which is meant for single cell isolation. We can handle biological objects in the range of 0.5 micrometer all the way to around 80 micrometers. So from tiny bacteria, nuclei to mammalian cells, and even big cells like cardiomyocytes, for example. And our second instrument, the SpheroONE that you can see here on the right, is meant to handle larger objects like spheroids, organoids, tumoroids, some tissue biopsy fragments as well. And so the combination of those two instruments enable researchers to handle all kinds of biological objects in that range from a few micrometers or even below all the way to 600 micrometers. Those instruments can come in various forms. So we have the standalone instruments, but we also tailored solutions depending on user needs. So we can integrate the instrument into a BSC, or those can come with plate shuttles in order to integrate them into a larger automation system with automated plate exchange, sample exchange, for example. So depending on user needs, we can leverage our knowledge and scientific expertise in bioautomation in order to answer customers' needs. Regarding our consumables, I will tell you more about that, but what we have developed typically are chips that enable researchers to automate and miniaturize single cell sample preparation on the CellenONE instrument for transcriptomics and proteomics. I will tell you more about that later. Those are the Cellen chip and proteochips. chips. And also in our laboratories in Lyon, we provide services to researchers that would like to answer a specific biological question at some point on these using those instruments and consumables. We can provide you some assistance with our services. So here's the content of my presentation. I will tell you first about the CellenONE, our platform for single cell isolation, tell you about how it works, and then touch the applications. And finally, I will finish with the second instrument, the SpheroONE, for 3D cellular models like spheroids and organoids. So let's start first with the CellenONE for single cell. So this is an all-in-one system that enables to do single cell selection, so sorting and isolation. And the same instrument also enables reagent dispensing in picoliter and nanoliter volumes, which is very useful for sample preparation if you want to prepare your cells for downstream analysis like DNA or RNA sequencing or protein analysis. And we also have some environmental controls, temperature and humidity, that allow us to perform on-deck incubation for those sample preparation protocols. And so in terms of applications, any kind of single cell omics we can do with that instrument. And it's also very suited for cell line development, where you want to grow you want to isolate single cells, have a proof of monoclonality for those cells, and then grow colonies or study antibody secretion from those cells, for example. So how does it work? It's based on 4 main pillars. The first one is an acoustic-based precision dispensing technology. The second is that we use imaging capabilities in order to isolate single cells. And then we have those environmental controls I told you about. And we have the capability to automate our workflows. So regarding the dispensing technology itself, how does it work? We have a proprietary technology, which consists in a glass capillary that you can see here on the right. And in that glass capillary, we can load some liquids or cell suspensions. And this glass capillary is surrounded by a piezoelectric ceramic. And so by applying a pulse, a voltage pulse to that piezoelectric ceramic, we have an acoustic wave that propagates throughout the liquid, and we generate a tiny droplet at the tip of the capillary. And by controlling the properties, the characteristics of this pulse, we can accurately determine the speed of droplet generation and also their size. So typically, we work around 300 picoliter per drop. We can work higher or with higher or smaller droplets, but typically it's in that range. The drop frequency can go up to 500 hertz, and those droplets are extremely reproducible. So it means that with the same voltage pulse and the same liquid properties, you will always have the same droplet volume, which is ejected. It's compatible with both aqueous and organic solutions, which is very interesting when you want to perform sample preparation protocols using various kinds of reagents. And so here is a short animation showing how the instrument works. Oops, let me run the video. So here is the instrument. And on the deck of the instrument, you can have positions for target and source plates in which you can uptake or dispense your cells or reagents. You can work as well with, various sizes of plates or the various other kind of chips or microscope slides. This is controlled in temperature and humidity so that your cells are in the best conditions. What we do is that we load a cell suspension into our glass capillary and we place it in front of a camera. And so here what happens is that the software of the instrument is going to do a mapping, a determination of what we call the ejection zone here in green, which corresponds to the volume of the next drop which will be ejected, and a small safety zone on top to take into account possible cell sedimentation. And so we are going to generate droplet and take a picture every time a droplet will be generated. When there is a single cell in the ejection zone, we move on top of the first target of the well and we dispense the cell. Here again, there is a second single cell. In the next droplet, we move to the plate and we dispense it. Come back to the camera. Here, there are no cells detected. We dispense the droplet. Here you see there is a doublet, so we don't want to dispense that to our plate. So we stay in front of the camera and we dispense it into that tiny recovery vial that allows us not to lose any sample, but to reprocess it afterwards. We also measure diameter, elongation, circularity, and all those elements along with the pictures of every cell is stored and can be accessed afterwards if you want to look at which cell was ejected into which well and what were those its properties. And so in terms of speed, we can go up to 40 cells per minute. What is really interesting with this technology is that as it's image-based and we have those pictures taken from the cells, it's really reliable, and we have a single cell accuracy up to 100%, which means that you will have no empty wells and no doublets or triplets, which is something that differentiates us from some other single cell technologies where it relies on Poisson distribution and you don't necessarily have only single cells in your wells or in your droplets. What is interesting as well is that the user can set some isolation parameters in terms of size and morphology of the cells. And we can also sort the cells according to fluorescence markers. We have 4-channel fluorescence system into the instrument. And so that allows people to select only the cells of interest. It is very gentle to the cells. We have a very low shear stress with that technology, so the cells remain highly viable after dispensing. As I said, we record images of all the isolated cells that you can link afterwards to your omics data, for example. And we can work from very small sample volumes, from just 1 microliter, which is very interesting for researchers working with rare samples, for example, patient biopsies with very limited material, where all the cells are precious and you don't want to lose any of them. We can work, as I said at the beginning, with typically any kind of cells. So with bacteria, nuclei, all kinds of microbes or mammalian cells, all the way to very big cardiomyocytes. We have customers that were able to isolate cardiomyocytes and do RNA and proteomics analysis on those. And we can analyze all of this in bright field and in fluorescence, depending on the user's needs. Something which is interesting as well is that we can work with highly heterogeneous samples at the beginning. So if you load cell suspension where there are very tiny cells and big cells in that same samples, we are able to isolate 100% single cells, all the small ones, all the big ones within the same sample, which is also something that not all single cell technologies allow their users to do. So yeah, as a summary of how it works, here you have the main elements that I mentioned. I won't repeat all of that. And on the right, you can see some pictures that were taken with the instrument or afterwards, showing single cell at day 0 and the growth of a cell colony from that single cell after 1 week, and various type of cells into our glass capillaries with the pictures that were taken. So various types of cell lines or mammalian cells, even microbes. So in terms of applications, as I said, there are 2 main groups. The first one is single cell omics, where you want to isolate your cells and then prepare them prior to analysis, whether it's DNA, RNA, or protein analysis, for example. And the other main group of application is the cell line development that we also call cloning very often, where you want to isolate single cells and do some gene editing or study antibody secretion and grow colonies from single cell in order to produce some of those antibodies, for example. So let's talk a bit about cell line development first and why this solution is interesting in that field. So the idea here is to complement or replace the very tedious and very manual protocols that are used in cell line development with manual methods, where you use, for example, limiting dilution, several rounds of limiting dilution. This is a process which is not very accurate, very tedious, and where in the end you have difficulties to identify the best monoclonal antibody producing cells, for example. And so with the CellenONE, as you have the assurance of isolating one and only one cell into the wells of your target plate, you can drastically reduce the time required for that process, limit human errors, and also have assurance of monoclonality. So this greatly accelerates the clone selection in cell line development processes. Something interesting is that we've shown that we had a very good clonal growth in any well plate format. You can see here on this chart that we tried with 96, 384, and 1536 well plates. In average, we got 95% of isolated colonies that gave rise to a viable colony afterwards, compared to around 25% with manual methods. So that's something very important in that field. We have a very gentle technology that allows the cell to grow after dispensing. We showed as well that we compared our technology to fax technologies, and we showed that in a single run, without reprocessing the sample on those hybridoma cells, we had a better cell recovery than the fax. And something more interested, because I mean, the FACS is a very established technology. It's kind of the gold standard for single cell experiments. We showed that we had a much better clonal outgrowth after dispensing, which means that our technology is much more gentle on the cells. We obtained 73% clonal outgrowth in this experiment compared with 14% only with the FACS. So something to consider if you want to maintain gene and protein expressions for omics experiments, or if you want to grow cell colonies afterwards. Also, what we did in the field of cell line development is that we were able to automate our solutions, because the pharmaceutical industry is really looking for solutions to fully automate those cell line development workflows. And so what we have in our portfolio is we have the standard CellenONE, with which you are able to do 10 or 20 plates a day. It's automated, but not in terms of plate exchange and sample exchange. The process is automated, but you have to manually change your sample and plates in between each run. And so what we did is that we developed the CellenONE HT platform that you see here on the right, which is fully automated with a software scheduler and allows the interfacing of the CellenONE with a plate shuttle on the side, with other instruments used in the cell line development workflow, like an automated incubator, a plate centrifuge, a barcode reader. And so with that, what we were able to do was to provide a solution to some big pharma customers that enable full automation, walk-away runs, overnight runs, with more than 10,000 clones prepared in a single run overnight, that enabled them to increase 10 to 100-fold the number of clones they were generating compared to other technologies. So that's really a success story for us. Our pharma customers are very happy with that solution. So if you are interested into single cell automation, that's something we can provide as well. And we can tailor our solutions to the user's needs. I now have a few words about microbiology. So working with single microbes, bacteria, or other kind of microorganisms. I won't be long on that one. It's just to tell you that this is something quite new with the CellenONE. We improved the technology over the past couple of years in order to be able to isolate very tiny objects, so very tiny bacteria, for example. And here you can see some pictures of those microbial cells in the glass capillary, and also afterwards isolated onto glass slides, for example, so very tiny cells, various types of microorganisms. This is something we can do. So if there is any interest for you to work on bacteria or other microorganisms, this is also something you can do with the CellenONE. I'll tell you about single cell genomics now. So here, starting with a success story as well, for people who want to analyze DNA from single cells. We have instruments placed at the British Columbia Cancer Center, and professors there and their teams developed a groundbreaking solution to miniaturize whole genome sequencing sample preparation on single cell. This method is named DLP Plus, for Direct Library Preparation Plus. And it's a method which is now used in routine at various cancer research centers in the world, MSKCC and New York Genome Center in New York. And it's mainly used to find new therapeutic strategies in hard-to-treat cancer patients. There are a couple of papers that were published in Nature and also in Cell over the past years, where they were mainly looking at clonal fitness and evolution of breast and ovarian cancer over time. And so this is now implemented worldwide in several cancer centers in the world. And so what this method enables, library preparation for whole genome sequencing, where you can study copy number variation, clustering, single nucleotide variation, as well as lineages. And the take-home message here is that with this method performed on the CellenONE, researchers are able to generate very high quality libraries without any preamplification. It works on any cells and nuclei, and in the end, it's at a very low reagent cost. What is stated in their publication is $0.25 per cell. And they have now at BCCRC sequenced almost 1 million single cells with that method. So nice papers to read if you are interested in whole genome sequencing on single cells. We also have solutions for single cell transcriptomics, where researchers want to analyze RNA from single cells. And so here we have developed a proprietary technology, a proprietary substrate that we call the CellenONE chip. It enables miniaturization of single cell omics workflows in those consumables that have the size of a standard microscope slides. And you can see that it's kind of 4 times mini 96 well plates on each of those CellenONE chips. So 384 wells per chip. And you can work in very tiny volumes in the nanoliter range and run up to 8 CellenONE chips per experiment on the CellenONE, which means sample preparation for a bit more than 3,000 single cells. So the CellenONE chip can come either blank, and you will put into the CellenONE, you will load into the CellenONE the reagents that are of interest for your own protocol and perform it. But we also develop some proprietary kits that allow for a specific workflow where the chip comes pre-filled with the reagents, and some other reagents come into tubes into the kits. And that's a complete solution. Here is an example with the 3' RNA sequencing kit. It enables you to focus only on your single cell isolation parts, and the kit does the rest with the right quantities of reagents. We provide the task and runs to run those protocols on the CellenONE. It generates really high quality data, and it's really ideal when you're working on small samples and raw cell analysis, which you cannot do with other single cell platforms, which are more suited for very high throughput, tens of thousands of cells per run. Here we can really work from just a few cells to a few thousands. So it's really a sweet spot for researchers working on samples in that range. And so that combination of CellenONE plus this CellenONE chip consumable solves most of the bottlenecks that you can meet when working in standard microwell plate technologies or with droplet microfluidic solutions, where it's not really flexible in terms of protocols you want to implement. You don't have any pictures of the cell, so you cannot link your omics data to the morphology of the cell. With the CellenONE and the CellenONE chip, this is something you can do. Any kind of protocol and correlation with the cell images. And importantly, working in very small volumes, which also limits the cost of reagents. A few words about the results with our 3' RNA sequencing kit. We did an experiment comparing a 2D culture of HEPA-RG, and we compared that with 3D spheroids of HEPA-RG. And we did an analysis of around 100 cells or 100 single cells per experiment, and we did a comparative gene expression study. And so what we were able to see was that we were able to differentiate among the different type of cells. We had the first cluster and the second cluster, which were corresponding to the 2D and to the 3D culture of cell. And we even had a third cluster showing the apoptotic markers, which are specific to the necrotic core of the spheroids. So in the end, the analysis of a low number of single cells, around 100 single cells, was enough to allow clustering and differential gene expression on those samples. So I think those are interesting results. We have an application note on that you can find on our website. We also show that those results correlate with other published work in liver tissue gene expression, and that was confirmed by our analysis. Another application for the CellenONE, which is really one of the hottest topics at the moment, is single-cell proteomics. And here we really have a unique solution in order to allow preparation of cells for protein analysis. So for those that are not very familiar with protein analysis, the difference with DNA or RNA is that you cannot amplify proteins. So it's really difficult to analyze proteins from a single cell because you have a very low amount of starting material. And until very recently, the mass spectrometers, the instruments that are used to analyze proteins, were not sensitive enough to work on single cell material. But since a few years, it's the case with mainly two technologies, the Bruker timsTOF SCP and the Orbitrap series from Thermo Fisher. Those have enough sensitivity to work on single cell material. But for that, you need a very clean sample preparation, not to lose any peptide across the sample preparation process. So here there are two competing but also complementary methods. You can either work in lab-free, preparing and analyzing the cells one by one, or you can work in a multiplexed workflow where it's a bit higher throughput because you will be able, a bit like barcoding when you want to analyze RNA, you can kind of barcode your peptides and know which cell it's coming from. And so you're able to multiplex up to 16 cells per run. So you identify more protein, but it's a bit more complex as well. And so what we did at Cellenion is that we developed two consumables named ProteoChip, one for the multiplex workflows and one for the lab-free workflows. And those consumables have a specific geometry that allows you interfacing with the CellenONE in order to dispense your single cells into the wells and also all the reagents necessary for the single cell proteomics sample preparation. For the multiplex, it also allows the pooling of the sample after barcoding with the TMTs. And then it's also directly interfaceable with the liquid chromatography instruments in order to inject your sample into the mass spectrometer. And in the end, both ProteoChips provide, together with the CellenONE, an end-to-end automated and miniaturized workflow for both multiplex and lab-free single-cell proteomics. So this is really, you start from a cell suspension, and then you have all the steps of the sample preparation, single-cell isolation, reagent dispensing for lysis, digestion of the peptide, labeling for the multiplex workflow. All the incubation steps happen into the CellenONE, and then the chip directly goes in the interface for the LC and injection into the DMS for data analysis. So this is something a work we did in collaboration with the IMP in Vienna Biocenter and also with Bruker and Thermo Fisher Scientific, which are providing the mass spectrometry instruments. So in the end, if you are interested in single-cell proteomics, we have a very unique solution that allows you to miniaturize and automate those sample preparation protocols and get very, very good data out of it. Going even further, we have a collaboration with PNNL in Washington, USA, where researchers in the team of Yingzhou were able to use the CellenONE in order to split RNA and proteins from the same single cell using a method they have developed. And they were able to do the first demonstration of unbiased analysis of both mRNA and protein from the same single cells. So I think this is really a groundbreaking work that will allow researchers to better understand cell and disease mechanisms and go beyond in the field of multi-omics. And there is a paper as well, which you can easily find on our website. So that's it for the overview of the single cell instrument and its applications, the CellenONE. I will now switch to the SpheroONE, which is our second instrument, which enables similar kind of work, but on larger biological particles like spheroids and organoids. So why do we want to work with spheroids and organoids? It's a paradigm shift from 2D to 3D in vitro models for disease modeling and drug testing. Because as you are probably aware, models in 3D are much more relevant to mimic an organ's function and behavior when compared to standard 2D cell culture. It's much more predictive for drug screening and various kind of research. And in the end, better recapitulates what will happen in the human body when being in contact with a drug, for example. And in the end, it will enable to complement and maybe even replace the use of animal models in biological research and pharma research. But the issue here is that it's very difficult to isolate single spheroids and organoids and to standardize drug screening. And so the SpheroONE is really a game changer in that field where 3D cellular models are used, because it's very similar to the CellenONE in the way it functions. So we have the same technology with a glass capillary placed in front of a camera in which we load not a cell suspension this time, but a spheroid and organoid suspension. And it really enables single spheroid organoid sorting, selection according to user-defined parameters, then isolation of single spheroids and organoids. And in the end, it's very useful to standardize the samples having the same size and shape of your spheroids and organoid in every well of your target plate in order to have more confidence into your drug screening workflows or any other kind of assay that you want to perform on the SpheroONE. An example to better explain that, where you want to perform a drug screening on 3D models, you want to plate spheroids into your well plate and to apply a drug gradient, so various drug concentrations depending on the columns of your well plate. And so usually people use manual methods, manual pipetting, but here the issue is that you will have a lack of control and very low reproducibility because you don't control the number of spheroids that you dispense, nor you cannot control their size either. And so if you have something very different from one well to the other, you have less confidence into your drug screening results. Whereas with the SpheroONE, you have first the ability to pre-strain the spheroid suspension to remove any debris that may bias your drug screening results in order to have a very clean spheroid suspension. And then you load that spheroid suspension into the SpheroONE instrument, and you are able to dispense one and only one spheroid per well with a predefined size or shape, and this greatly enhances the homogeneity and reproducibility of the experiments. So with that instrument, you can dispense a single spheroid per well. you can define which ones are of interest and you want to isolate, which enables the user to start from a very heterogeneous spheroid suspension and end up with something very homogeneous. You are able to remove debris before the instrument. And in the end, you have something that is also very useful for organoid generation, where you can sort your 3D aggregates before differentiation in order to optimize the yield. Something I did not mention is that we can also pre-fill the plates with matrigel and dispense organoids directly into matrigel, because our plates are temperature controlled, and this greatly helps into those experiments in order to have a high yield of differentiation. So this is it for my talk. On this last slide, you can see examples of customers that are already using our technology, trusting our technology all over the world. It's not an exhaustive list, but those are some examples of leading research centers in the world using our instruments. And with that, I want to thank you for your attention. I will be glad now to take any question you might have on our technologies, and you can also afterwards reach out either to Ricardo from Axela, who is distributing our products in your country, or you can reach out directly to me. Here are our contact information. But if you have any question right now, I'm happy to take those.
Thank you, Sebastian, for the great talk. And I really hope that there are a lot of questions, because, yeah, these are just fantastic instruments. If I could, I would have both in my laboratory. So I really hope that the audience would have a lot of interesting questions. So you can just unmute yourself and ask, because we have the possibility. There is no need to ask in the chat box. So feel free to ask anything. Seems like no questions, surprisingly. Okay.
So just to conclude, I would like to say that if you are interested into publications, application notes, more information about our products, you have a lot of additional contents on the Cellenion website. You can go there and look for the information which is relevant to your application, and don't hesitate to reach out to us if you have any follow-up question after that.
Actually, I have one.
Oh, sure.
I'm Dagmar, also from Axela, so it will be just a quick question. Maybe I missed it. What's the sorting speed of the SpheroONE?
Yes, I did not mention it actually. So it's a bit slower than the CellenONE. With the CellenONE for single cells, we can go up to 40 cells per minute. With the SpheroONE at the moment, we are more around 20 spheroids per minute. So it means 5 minutes in optimal condition to fill 96 well plates, depending on the parameters that you set for the morphology, so size, shape, etc. If those aggregates of interest are a little bit more rare in your sample, it can be a bit longer, so 8-10 minutes per 96 well plate. It really depends on the starting sample and what are the spheroids of interest that you want to isolate, but up to 20 spheroids per minute.
Thank you.
I have also one question. So for SpheroONE, which would be the necessary space for already existing laboratory just to place it inside?
So I try not to say any mistake about the size. I think it's something around, for the footprint on the ground is like 70 by 70 centimeters, but you also have, let me show you again. You have this arm screen as well, which is something like 65 centimeters. So let's say the width is 130, something like that, 70 centimeters in depth, and the height of the instrument is 160 or 80. So yeah, let's say 2 meters square.
Okay. Thank you.
You're welcome. You can find the exact dimensions on the brochure, which is on our website.
Yes,
if you want the
sure. I
exact
was just
information.
curious. Yeah. No, the idea is I was curious if it's necessary to have more space except the instrument itself.
No, just
Mhm.
that.
Okay.
Or no, if you take, actually, if you want the temperature and humidity control, there are some additional accessories, but that can easily fit next to the SpheroONE instrument, so it doesn't take much more space.
Mhm. Okay.
Hello, I would like to ask also, I'm Csaba from Hungary.
Mhm.
Is it allowed to ask the presentation? Because I have got some customers who couldn't participate in this webinar because they had got any other education task, and I would like to share this presentation with these researchers.
I can answer to that, Csaba. So yes, there is, you anticipate me, there will be the recording, which we will share on YouTube in about a week. In about a week, so everyone will have access to that for sure.
Thank you, Ricardo.
So if there is no more question, thank you once again, Sebastian, for this nice talk and for your time today. And thanks to every participant. And of course, you will have a chance to review the recording, and feel free to get in touch with us for more questions.
Thank you.
Thank you,
Thank you and goodbye, everyone.
Have a good
nice
time,
day.
everyone.
Bye.
Merci, Sebastian. Bye.